bachelor's from University of Texas, Austin, and then a master's and PhD from MIT. Between 95 and 97, Dr. Rogers was a junior fellow at Harvard University Society of Fellows. And he also served as the founder director of Active Impulse Systems, a company that commercialized technology developed during his PhD. Then he joined the Bell Labs as a member of the technical staff in quantum matter physics in 97 and served as the director of this department from 2000 to 2002. He is currently the Swanlund Chair Professor at our university, which is the highest professorship that we have on campus, I think. There is a long list of honors that Professor Rogers has. We could spend the whole hour reading these, but let me just tell you a few. He received in 2009 the MacArthur Fellow Award, which, as you know, that is from known as the Genius Award. In 2011, he became the member of the National Academy of Engineering. He also won the Lemelson MIT Prize in 2011. And I'm skipping through a lot of this. So last year, he received the American Ingenuity Award from the Smithsonian Magazine. Finally, this year, he became a member of the Academy of Arts and Sciences. John, thank you so much for accepting our invitation.
Okay, great. Thanks for that kind introduction. So it's a pleasure to be here. I'll tell you about some of the things that we've been doing in our group, kind of in the area of tools for neuroscience, I would say. And in particular, a specific class of technology that we think could have utility in this emerging field of optogenetics, which you're probably familiar with. So my background is in material science and engineering. We do a lot of related things, but I think the core value we can bring to a field like this in biomedical research is new materials, new devices that allow you to do more sophisticated things than would otherwise be possible. So that's kind of the context and the way we're hoping to make a contribution into this field. So I'll provide a little bit of an overview and why we're interested in this area, what we think represents some useful classes of materials, how to build devices with them, and then, like I said, I'll really focus specifically on devices that allow you to do pretty sophisticated optogenetic studies by use of brain injectable light-emitting diodes and coupling of those to wireless power harvesting and control systems. So I think, kind of in a broader sense, what we have spent some time on over the last several years is, asking the question, what could one do to more readily bring to bear on problems of human health and research in biomedicine, the kind of sophisticated semiconductor device technology that's all around us in the form of consumer, industrial, and military systems, where spectacular levels of function and integration and cost and reliability can be achieved with devices that you essentially etch on the surfaces of semiconductor wafers, whether it's silicon or gallium arsenide, gallium nitride. These are very, very powerful material platforms for doing all sorts of stuff. In terms of integration with the body or with biological systems, however, this format of material is not particularly useful because the geometry and the mechanical properties are dramatically mismatched, with the soft textured surfaces of biological systems. And so that creates a set of challenges and associated opportunities in the development of ideas in materials and mechanics and in manufacturing science that would allow you to sort of reformulate this kind of technology as it exists today into forms that could very naturally and intimately integrate with biology in ways that would otherwise be impossible. And so that's what we've aimed to do in this area. And one strategy might be to look at this type of device platform and say that it's never going to work and that you need, therefore, to develop alternative classes of semiconductors, polymers or small molecules or graphene-based semiconductors, carbon nanotubes. I think those strategies are appealing. We spend a lot of time on them, but at the same time, you really have to acknowledge that this is a very, very well developed class of material, and there's tremendous depth of scientific and engineering capabilities that have been built up around it over the last half century of research in integrated circuits and optoelectronics. So instead of trying to reinvent that kind of material, we have focused instead on changing the way that it's used. And I'll just step you through a couple of really simple concepts to give you an idea of what we're trying to accomplish and some strategies that work well for us. So if you think about this format of material, it has the properties that I just mentioned. It's very planar, flat. It's rigid and brittle. If you drop it on the ground, it'll shatter into a million pieces. If you try to wrap it around a body part, it will crack. And that's partly because of the intrinsic materials properties, but it's also a consequence of the fact that the wafers are pretty thick. So maybe a ½ millimeter thick or a millimeter thick, depending on the diameter. And that renders a relatively large bending stiffness. So the bending stiffness scales like the product of the modulus of the material. That's sort of intrinsic. You're not going to change that. But it also depends on the cube of the thickness. So if you plot the bending stiffness as a function of thickness, you see that you have a value for silicon that's around 10 Newton meters for a wafer scale thickness, so maybe a millimeter. But because of that cubic scaling, this is sort of a log-log plot, the bending stiffness decreases by many orders of magnitude as you reduce the thickness from a millimeter to, say, 10 or 100 nanometers. And so in this regime, the bending stiffness is maybe 10 femtonewton meters, and that's 15 orders of magnitude, then decrease in bending stiffness just through geometry control. And this is just linear elastic Newtonian mechanics, but that huge 15 order magnitude change in bending stiffness has a qualitative impact on the way you think about the material. So it's no longer a rigid plate, but it's a thin floppy noodle. And you can see it here. This is a 20 nanometer thick sheet of monocrystalline silicon. So that's sort of interesting. The other consequence of small thickness is that for a given bend radius, the peak strains induced in the material scale down linearly with thickness. So R is the radius of curvature, H is the thickness again. So not only does the material become floppy in the sense of a very, very tiny bending stiffness, but it also becomes very flexible. You can bend it to a very tight radius of curvature before you reach thresholds of strain that would crack the material. So, so those are useful things to think about. Initial instinct may be, okay, well, we'll replace the wafer with this thin sheet of silicon and then you'll build the devices that way. That doesn't work either, though, because this is very thin and floppy and flexible, but it's also very fragile. So you have to think about it not as a device platform, but as a building block material for devices that you might construct on a sheet of plastic or a piece of rubber. And in those cases, you have to grapple with a problem in heterogeneous integration. So how do you put silicon on silicone and manage that interface so that it remains robustly adhered during practical use? And there, that would be daunting. If you were taking a chip of silicon and trying to glue it to a piece of rubber, you'd have a hard time doing that. But if you think about the mechanics of interface failure between two joined dissimilar materials, the propensity for cracks to form scales with the energy release rate G. And for the case of thermally driven mismatch strain, the expression kind of looks like this. But for other ways that the crack can open up, it also scales linearly with thickness. So again, as you go from a wafer to a nano membrane, the energy release rate scales down by many orders of magnitude, thereby allowing you to achieve robust bonding interfaces, even between radically different materials like silicone and silicon. So if you look at a plate of silicon, for example, very thin, you can print it onto a micromachined ridge on a piece of plastic, and it will remain in this cantilevered state just due to van der Waals interactions. It's partly due to this linear downscaling of the energy release rate that you can do something like that. So, very flexible, floppy, and sticky is what happens when you make things very thin. So then the next question, if those kind of mechanics are appealing, again, for doing biointegration, how do you create those membranes in the first place? And one strategy might be say, okay, I'm going to develop some synthetic techniques to grow those materials from, let's say, vapor phase precursors. And you might think about doing that, but again, you'd probably be well served by acknowledging the fact that people already know how to grow these classes of materials in bulk form extremely well, and in supported thin films. So for example, you can create these membranes in this kind of strategy. You do MOCVD growth of gallium arsenide and aluminum arsenide in alternating layers. And that's interesting because when you etch down through this stack, you immerse it in HF, you will selectively eliminate the aluminum arsenide, lifting off the surface of the growth wafer bulk quantities, essentially these gallium arsenide nano membranes. And then you can reuse the wafer. So this provides a really easy and powerful route to extremely high quality, very thin sheets of semiconductor materials. And you can do this quite well. We work with Shuling Lee here in electrical engineering. There's the 40-layer stack, gallium arsenide 200 nanometers thick, aluminum arsenide sacrificial interlayers, again, 20 nanometers thick, etch down, eliminate the aluminum arsenide, lift off a lot of gallium arsenide nano membranes. And you can do this in heterogeneous stacks also. These are just uniform layers of gallium arsenide. You can change the doping constructs to create solar cells, sacrificially separated from, let's see, near infrared detectors, MESFETs. You can do sort of heterogeneous collections of materials for different classes of devices the same way. And this is what it looks like if you lift it off and then deposit it down onto filter paper. So you can do this, and you can do it, in large quantities in strategies that are fully aligned with mainstream industrial approaches for growing materials and, and implementing them in devices. So that's mechanics, that's materials. The next question is manufacturing. How do you move these things around and get them into arrayed configurations such that you can establish electrical interconnects and build systems that make sense? This is not a good starting point for that because you have to sort of disentangle all these things, move them around. You can't do it very easily because they're very, very thin, fragile, and very small. So how you do that becomes another topic of research. This is a way that has worked well for us. So you can take these wafers, you create these little platelets, undercut etch them so they're completely released from the underlying wafer except at strategic anchor points. So maybe at their corners you define pieces of photoresist so it keeps them in their lithographically defined locations. If you do that, then you can think about the wafer as an inking pad. For a stamping process, a printing process, use a soft elastomer stamp with relief features on its surface, bring it into contact, tacky surface of the stamp adheres sufficiently strongly to these released nanomembranes that when you peel them back, you fracture the anchors, ink up the stamp, index the stamp over to a target substrate, bring it down, print them, and then do this over and over again. So you can take very dense collection of very tiny, very thin material elements and then spread them out wherever you want them on any kind of substrate. And because you're doing this at room temperature, you have a lot of choices on substrate because you don't need to accommodate the growth conditions that are needed to form the materials to begin with. And so there are a lot of fundamental aspects around how you control adhesion between the stamps and these elements because you need to be strong enough to allow efficient inking, but you need to allow them to get off the stamp so you can print them down. So it really requires switchable adhesion strength on the surface of a soft material. There are a lot of ways to do that. I won't get into them now, but just say that can work. You can build tools like that to do it in an automated way. You can take arrays of these very tiny things in dense configuration and spread them out any way you want just by programming up the printer in an appropriate way. So here you're going from 50 by 50 micron nanomembranes on a 70 micron pitch, and then you can spread them out, 0.7 millimeters, 2.8, and a little over 5 millimeters just by programming the stamp. And that turns out to be powerful, and I'll show you how that works in the context of optogenetics in a few minutes. So here's an example. Automatic printing of gallium arsenide membranes in a low aerial configuration on a sheet of plastic that we subsequently wrapped around cylindrical glass support. In this case, is 1600, 100% yield. You see the bending and the adhesion mechanics that I mentioned to you before. Gallium arsenide is extremely fragile, but it's very thin. You're on a thin substrate, then you can bend it without cracking these things. And furthermore, it's sticky interface. So these elements don't just pop off as soon as you bend the substrate. So all those things come into play and allow you to do that kind of thing. Okay? So if you can put semiconductors wherever you want them on any kind of substrate, then you can start playing games with mesh designs, buckling mechanics, unusual materials that allow you to build integrated optoelectronic systems that you can integrate with the surfaces of biological systems, brain, heart, and we've done a lot in skin. And this allows you to do levels of fidelity and precision in, let's say, surface potential mapping, both in the brain and in the heart that would otherwise be impossible if you were just using sort of bulk electrodes without active multiplexing or local amplification. So you can bring the full power of wafer-based optoelectronics to bear on an organ system like the brain or the heart. Here, sitting on the surface, sort of 2D roughly, but conforming to the sulci and the texture of the brain. In this case, we created the electronics on a membrane that fully envelops the heart, so specifically designed in the shape of the heart itself by optical scanning and 3D printing techniques. So I won't tell you anything about this. We just mentioned that this provides a great non-invasive way to integrate with surfaces. But these are 3D organs, so you can only do so much on the surface. Like you might want to have access to the depth, and that's a much harder problem, but one that can be addressed with some of the ideas that I have talked to you about so far. And I'll focus the rest of my talk on that topic, plus the fabrication and use of very, very tiny, thin LEDs. And you see one of them right there, emitting in the blue, this indium gallium nitride. Let me tell you some more of the details about how you make those devices, and then I'll show you how you implant them down into the brain. So in this case, instead of using aluminum arsenide as a sacrificial release layer, what we do is we grow indium gallium nitride on a silicon wafer that has a 1-1-1 orientation. You can do that. You can create high quality InGaN that way. And then the silicon wafer with that orientation has the appealing property that it etches anisotropically in a solution bath of potassium hydroxide. In particular, the etching rate along this crystallographic direction is about 500 times faster than it is along that direction or down into the depth. And so if you orient your devices that you create in the InGaN material grown on this wafer in this fashion, such you have anchor bars running along this direction and this direction, then when you etch down to delineate the lateral dimensions of the devices, you expose a little bit of the silicon, you immerse that in potassium hydroxide, and then you undercut just along this direction. And so the devices then can be configured with little anchor points at the corners, and those provide tethers to these bars such that when these devices are completely undercut etched, they're sitting in a freely suspended configuration and they're ready for printing. So here's one of these devices, little anchor bars, come in with a stamp, pluck that off, fracture the bars, and then you can print the devices down wherever you want. So this allows you to make devices that are exceptionally small or big, whatever you want. It's lithographically defined dimensions, so you can go from sort of a conventional LED size in terms of lateral dimensions, 1 by 1 millimeters. The thickness is only about 5 microns, and that's about factor of 20 to 50 times thinner than a conventional device, because the way conventional devices are built is use a wafer saw. So you slice up the wafer, so you carry the active material, but you retain that silicon wafer substrate, so much, much thicker. But you can do large or all the way down to small. So this is about as small as we can do with the academic facility, but in principle you go much, much smaller than this if you want. It's about 25 microns on a side. If you have better lithography capabilities, you can go smaller. So small will be useful for a variety of reasons that I'll describe in a few minutes. Thin is critically important because it allows you to do planar processing of the contacts and the interconnects. So here's one of these thin devices. You can just spin cast a polymer on top of it to sort of planarize the thing, open up holes to the contacts at the corners, and then you can do lithographic edge over metal deposition to define the interconnects, and you can make connected arrays of LEDs very easily without doing the kind of wafer bonding that's traditionally required. So the upshot is that this kind of inorganic LED technology begins to look a lot like organic LEDs in the sense that they're very thin, they're compatible with planar processing, and you can develop large-scale arrays of them on a planar substrate. And that's a lot different than what you can do with LEDs today, where you dice up the wafer, you end up with these big chunky blocks, and then you move those around with a robot, you put them in cans, and then you do wire bonding to form pairs of terminals to each individual device, and then you take those things and you tile them up and you make giant macro scale displays, but in a way that does not downscale in size or upscale in number of pixel elements very easily. The other advantage, I'll come back to this in a second, of very tiny small LEDs is the ability to do passive thermal management. So because the surface area to volume ratio is very high, the passive rates of cooling are very high, number one. And then number two, the electrical interconnects themselves have sufficient thermal mass that they suck heat away from the devices themselves. And so if you compute surface temperature as a function of LED size or interconnect thickness, you can see the tremendous benefits in going down in size and up in interconnect thickness. You can operate these devices at very, very low operating temperatures using those two concepts. And that's extremely important for integrating LEDs with biology, because you have a very narrow thermal budget in the context of living cells. So here's an example, arrays of these devices all interconnected. You can put phosphors on, you can do any color you want, start with the blue, down convert, and you can create sort of lighting bands. And we have a company that's doing commercialization of that. I won't mention it here. But the relevance to biology is, I think, multi-faceted in terms of opportunities. But I'll just focus on optogenetics, which, as you may know, is a very powerful and fairly recently developed technique in neuroscience in which you use genetic modification to create channel rhodopsin in the membranes of specific neurons. And if you do that in the right way, then you create some optical sensitivity to their function, so you can optically excite or inhibit their activity. And that's important because it provides a much higher level of precision than what you can do with electrical stimulation, which is not cell specific. And so this is creating huge opportunities in doing new neuroscience studies, behavioral studies, and understanding how brain structure and function controls behaviors. And that's pretty exciting. But everything that's being done right now is driven by neuroscientists who aren't very good with materials and engineering. And so this is the kind of thing that has dominated the field. In fact, the only platform that's really been used for any substantial work in this area. It involves a optical fiber that's implanted down into the brain on one end, and then the other end connects to what is not visible in this image, a separate box that's providing the light source, typically laser. And you have some dental cement and a fixture here. So in this way, you can certainly introduce light down into the brain. But if you're trying to study behavior, you have to wonder about what this tether is doing in terms of restricting that natural behavior. And then furthermore, you can't really allow the animal to explore a very complex environment because they get tangled up with the fiber optics. And likewise, social interactions become very difficult because everybody's tethered and getting tangled with one another. So that's the landscape that we observed when we got involved in this in late 2011. And our idea was to say, throw out the laser, throw out the fiber optic cable, and put active LEDs down into the brain and use wireless control and harvesting strategies to enable really free moving animals to be modulated by optogenetics. So use the ideas I just described you. Very tiny LEDs, almost cellular scale in their dimensions compared to fiber optic. This is kind of what it looks like. And then mount them on ultra thin filaments of polymer. So narrow strips, very thin polymer substrates, do the transfer printing, do the edge over metallization and interconnect, and form little arrays of LEDs such that the entire polymer filament here is only maybe 5-6 microns thick. And that tiny thickness, smaller than the dimensions of an individual cell, it allows you to do not only LEDs, but you can stack up other devices on top or beneath these LEDs to create multifunctional systems. So you can do LEDs, you can do photodetectors, little thin silicon pin diodes. You can do electrodes for measuring or stimulating electrically. You can do heaters or temperature gauges down here. So thermal management is very important, as I'll highlight in a few minutes. And so stack these things up. And at the end of the day, you're maybe at 25 microns. And that's good in the sense that it involves minimal displacement of tissue during injection. But what it means is because of the bending stiffness scaling with the cube of the thickness, it doesn't have enough rigidity to actually penetrate the brain tissue. So what we do is we use a releasable then microneedle that's much thicker, lithographically defined in a polymer to match the shape of these devices, and then adhered to it with a bioresorbable glue. So you inject the whole thing down and then you release the devices by taking the needle out. Here's what they look like without that releasable injection needle, just to give you a sense of the dimensions and the flexibility. This is one of these devices threaded through the eye of a sewing needle and then wrapped around its shaft to give you a sense of the flexibility and the geometry here. So here's what it looks like injected down into the brain right before the injection needle is still mounted. Plunge it down into the brain with a controlled, computer-controlled stage so you can locate these LEDs specifically in targeted areas of the brain. Once it's down there, we introduce artificial cerebrospinal fluid. That dissolves the bioresorbable glue. It's a silk fibrin material in this case, biocompatible. Once the needle is then physically released by that dissolution process, you can pull it back out. So then you just have LEDs embedded down in the brain and then electrical contacts emerging from the surface of the brain for connection to external control systems. So the very small geometries of these devices greatly minimize gliosis and other kinds of unwanted biological responses. So you can compare metal cannulas, obviously the worst. This is the fiber optic cable here. This is the LED with that releasable microneedle injection system still in place, and this is what it looks like. You pull it out. So compared to all of these reference points, the ultra thin multifunction LED system is providing the least invasive type of implant. It also provides other opportunities in controlling the directionality of the light. So if you have a fiber optic, you just have a cone of light coming out the tip end. But with the LEDs, you actually get light emerging from the sides, or you could get it down to the end. And you can put reflectors, for example, to control where it's going one side or the other. You can also do multi-colors. So showed you blue, you can do red. You can do red and blue. You can do green. Pretty much whatever you want and need in terms of your optogenetic sensitivity. So the devices are now in the brain, and you think about it as like a light bulb in the brain. That could be a good thing, could be a bad thing. You have to recognize that the brain can't tolerate temperature changes more than a couple of degrees before bad things start to happen. And if you think about that in the context of an LED flashlight or headlamps, you quickly realize that thermal management is going to be an interesting thing to think deeply about. And we spent a lot of time around thermal management. And you can do a number of different studies. Here's an example of LEDs implanted into a brain that's been extracted from an animal so that we can do IR imaging. And we can operate that LED at different repetition rates and then map the temperature. So the peak temperature is 37 up to 37.5 roughly, even at 20 hertz, not too bad. When you implant these things down into the brain, you can co-locate one of these high precision temperature sensors right next to the LED and then measure it in vivo. And that's what's shown here. This is the change in temperature measured at the position of the LEDs as a function of time immediately after they're turned on and then turned off again. And what you see here is, again, the temperature change is very small. So why is that? It's sort of a couple things. One is that the LEDs are very tiny. Surface area to volume ratios are high, so you get a good passive rate of thermal diffusion, as I mentioned to you previously. The other thing is that for optogenetic stimulation, you only need to operate these devices at very low duty cycles. This is 20 hertz repetition rate, but then the devices are only on for about a millisecond during that time. So the duty cycle is low. That's extremely helpful. And then the other thing is in the living system particularly, and you can see the delta T in the living system is much smaller than that in this explanted brain sample. And that's because the blood flow, the circulation through the vasculature next to the LED is actually heat sinks. So it's pulling heat away from the devices. So even at the highest repetition rate, the peak temperatures are less than or about comparable to about 1/10 of a degree. So that's more than a factor of 10 smaller than it needs to be in order to avoid adverse effect of the heating. Okay, so this is what it looks like then. Have LEDs on this thin filament implanted down into the brain, and then we have a wireless energy harvester here. So it's a thin flexible PCB technology. We use a 900 megahertz ceramic antenna. That mounts on the outside of the head. It plugs into a releasable connector here so that we can plug in the module when we're doing optogenetic studies and then take it off when those studies aren't ongoing. So this allows us to deliver power to the LEDs, and then by simple extension allows us to turn the LEDs on and off using a patch antenna that we put in a corner of the room that has the cages for the animals. So we can do one or many animals, and we've removed entirely the any kind of tether. So this is what it looks like. So these are the animals. This is the power harvesting module on the outside. It's a little bit bulky. I'll show you much smaller ones in a second, but we are unable to see any measurable difference in activity levels of mice that have these devices and those that don't. So it doesn't seem to affect their behaviors to the extent that we can measure that. Here's a simple optogenetics experiment to illustrate that the devices are working. So in this case, the mouse is genetically modified to allow us to stimulate neural circuits that produce dopamine upon light exposure. And the dopamine creates a pleasure response in the animal, and we can use that pleasure response then to train complex behaviors without physical reward. So what we're doing here is allowing the mouse to explore this Y maze habitat, and whenever the mouse comes into this region of the Y maze, we illuminate the brain, activate the RF antenna, light up the LEDs. That creates dopamine, and it creates an affinity in the animal for this part of his world. And so if you do that enough times, then the animal doesn't want to go anywhere else. It's just hanging out right there all the time. And this is just a simple demonstration that the technology works. This kind of optogenetics study has been done in the past with fiber optic cables because you can do that. It's a very simple environment. It's not a social experiment. But this is a good starting point for us to illustrate that the device works. So here's what it looks like. You do those kind of experiments. If you do not train the animal, they like the ends of these corridors, but they like each one with equal affinity. But if you do the training, then even when you're not operating the LEDs, they stay in that part of the maze. So that works. So what are we doing now? We're doing two things: working on advanced technology, and then working with our collaborators to use the existing technology to do more advanced optogenetic studies than had been previously possible. So we supply devices now to probably a dozen groups who are interested in their use. We have many more folks who would like devices, but that's maxing out our bandwidth and just building these things roughly. So we're doing a lot in that domain. I won't talk to you about any of that, as some of it's still ongoing. But I'll tell you a few things about the technology advancements. So there we're focused on two capabilities, really reducing significantly the size of the harvester to the point that we can actually implant the harvester subdermally. We're doing that. The other thing we're doing is we're creating additional functionality in the injected needle structure. So we now have devices that allow you to inject not only light but fluids. So you can do drug delivery, and you can also do extraction of interstitial fluids. I won't tell you anything about that. Those devices are working, and we have animal model results, but I didn't have time to make the slides. I'll show you just about the harvester. So this particular device was designed for operation at 900 megahertz. One simple way to reduce the size is just to go from 900 to 2.5 gigahertz. And immediately everything scales down because of the shorter wavelength in that regime and allows us to get the whole device now under the skin. So it's about a factor of 60 smaller in dimensions, about a factor of 20 reduced in weight. And so at this point it's just a tiny fraction of the weight of the animal itself, about 1,000 times smaller than the weight of the animal. So this is what it looks like then when you do that. So you open up the intracranial space, you inject the devices, and then the harvester looks like this. So we encapsulate it in a thin layer of silicone, essentially, to prevent ingress of body fluids, and then it's small enough to go under the skin, and then we can suture it up when done like that. And because the antenna is close enough to the surface of the skin, losses associated with absorption into biofluids are manageable. So we can get RF radiation through the skin without much problem. So this is one of the first animals that we implanted with these devices. So the LEDs are down into the brain. LEDs terminate at a harvester, which is sitting right here. And this is a gel placed on top of the wound to avoid infection. So now we can do things like this. And in fact, you can now take these kinds of devices and use them in optogenetic studies of other parts of the nervous system. And in particular, you can use optogenetics to modulate pain response by stimulating or inhibiting activity of the sciatic nerve. So you can take these devices, you can put them in the hind leg, and we're doing that kind of work as well. So that's really all I had to say. The work itself relies on close collaborations with senior PIs at Northwestern on thermal modeling and theory around that. Michael Brukas is our collaborator in optogenetics for studies of the brain, and we work with Bob Giroux, also at Washington University, on these studies of pain modulation. The students, 3 very good postdocs, Taeil Kim, who's now a professor at SKKU, Gunchul Shin, still here building the LEDs, and Sung-Il Park from Stanford is focused on the RF system. So with that, I'd like to thank you for your attention. I'd be happy to answer questions if you have any. Yeah, so we have had these devices implanted in animals for as long as a year. We haven't seen anything over that time frame. But we're not aiming for 10 or 20 years. I mean, that could be a relevant target to think about in the context of deep brain stimulation or things you might want to do with humans outside of optogenetics, just using the injectable electronics platform to do some things along those lines. I think those are interesting directions, but we really haven't push the studies to that degree. So for this work, it doesn't seem to be a problem. Yeah. Yeah.
So you have the device that can do stimulation.
Yeah. Yeah.
Do you think there's a potential to do also imaging, so you can monitor?
Yeah, so we have an NIH program not to do full imaging. I think that would be the ultimate thing. We're working with Steve and trying to think about how that might happen. But in the meantime, we have a separate program with folks at NIH that's designed to allow fluorescent measurement of overall brain activity in the vicinity of the region that you're doing optogenetic stimulation. So we have, in that case, LED to do the optogenetic stimulation. Then we have another LED color to stimulate the fluorescence, and then we have a narrow band optical detector. So it's not imaging, it's like single point measurement. But I think maybe that's a step in that direction. I mean, it would be awesome if you could do full imaging, but you have bandwidth on the communications back out. It's quite a bit beyond what we've done so far. And I think just the global fluorescence measurement is a good first step to think about something more ambitious. Yeah.
Other question? If not, let's thank our speaker one more time. Thank you.
Yeah.